Hi, everyone, and welcome to another episode of the Biotech Blueprint podcast. I'm your host, Katarina Rosnick, and today I'm joined by Miro Gasparek, CEO and co-founder of Sensible Bio, and Krishna Modramgari, the principal computational scientist at Sensible. So Sensible Bio has been making waves lately with major partnerships and rapid advances in cell-based mRNA manufacturing. Welcome, Mira and Krishna. Before we dive in, can you each give a quick intro about who you are and what you focus on at Sensible?
Katarina, thank you very much. It's really great to be here with you. So, Miro, CEO and co-founder of Sensible, as you mentioned. My background is actually in biomedical engineering. I was hopelessly torn apart between theoretical astrophysics and medicine in a high school, so ended up going for a biomedical engineering, which I thought is a good blend of both, and was doing a bunch of research throughout my undergrad at Imperial, Caltech, Stanford. Very quickly got engaged in my own entrepreneurial projects, and when I embarked on my doctoral studies in control theory applied to synthetic biology at Oxford, I already knew I wanted to be an entrepreneur, and inspired by a global pandemic and some of my other work on biosecurity at Stanford or with WHO, I actually ended up co-founding Sensible Biotechnologies with my co-founder and our CSO, Marian Kupchulak, with whom we met during our doctoral studies at Oxford.
So was COVID kind of the thing that inspired you to start the company?
It didn't inspire me to start a company. I wanted to start a company nevertheless, but it inspired us to pick up the problem that we are solving, really building the world's first fully integrated mRNA therapeutic engine. Because I had the opportunity to see firsthand from my work with Slovak government, WHO, or my kind of biosecurity research with Megan Palmer at Stanford, how important mRNA can be, and not just for infectious diseases, but beyond. And I started thinking about the challenges this technology has together with Marian, and we thought that perhaps there is more dogmas in mRNA technology to be addressed. But I'm sure we will get to this a bit later.
Yeah.
Yeah. Thanks, Miro. Yeah, I'm Krishna. I'm the principal computational scientist at Sensible. My background has been in, computational biology and bioinformatics. I started straight out of undergraduate. I've been working in this field for 15 years now, I think. Yeah, and it's like I kind of just fell through it. I don't know. I never really predicted it to be a career, much less a lifetime. I was always in my lab, always the guy good with computers, and that kind of that kind of started from my undergraduate. So I'm originally from Michigan. Went to school there at the University of Michigan, and I kind of come out here for a PhD in Ireland, where my background is in ML-based understanding of like looking at optimizing biomanufacturing, and so that's the connection Mira and I have, and so we're both very passionate about this, and we're seeing kind of like it's a landmark event right now where things are changing across the industry. So it's great place to be and great time to be here.
Yeah, thank you. Thanks to you both. So Mira, you mentioned you worked in policy in global health and now in biotech. If we can get a little bit to sort of the mRNA side and what felt most limiting about it, because you kind of touched on it a little bit. And when did it become clear to you that solving this needed to happen through like a startup like Sensible?
So when I was thinking about mRNA technology, it was really a journey. We were really problem driven, and Maren and I originally thought that the biggest challenge of mRNA technology is really the cost and scale at which we can produce it. Because, like, think back to the COVID pandemic, and I'm simplifying this a bit, but once the sequence of the virus of the SARS-CoV-2 was available, people really were able to design the sequence of a prospective vaccine literally over the weekend. And then when everything was working at its top speed, people were able to run the clinical trials in the course of months because regulators were receptive and just everybody prioritized it. But what became a bottleneck was scale up and manufacturing when really the pandemic was over before we could make enough vaccines to vaccinate the people. And I would say importantly, the pandemic of COVID was over before the pandemic of misinformation had started. And I think that this was actually a challenge, right? We couldn't get people or enough people vaccinated before all of this misinformation around the pandemic has spread, and before like all this lack of trust came up. And so originally we thought the biggest challenge is really scale up and associated cost, because turns out that mRNA is very expensive to make. And however, as we were kind of talking to the leaders in the field, and as we were exploring this deeper, we were really realizing that where the biggest missed value is really in mRNA therapeutics, in the opportunity to make enzyme replacement therapies, new cancer immunotherapies, gene medicines, cardiovascular treatments. And this is where none of that has been enabled, although that was, I think, the original idea of mRNA. And I like to say, paraphrasing this quote, as some venture capitalists say, is like, we wanted flying cars, we got 140 characters. And I like to say we wanted tens of mRNA therapeutics, and we got 2 infectious disease vaccines, really, COVID and RSV. And there are hundreds of clinical trials, but we have never gotten any other mRNA therapy approved, and hopefully they will get. And we started thinking, what if the biggest challenge is really the biggest untouched dogma? And the biggest untouched dogma of mRNA is really the mRNA manufacturing and the way we design mRNA for that manufacturing.
Yeah,
That's how it
so
started.
yeah, that's really cool. I know your team is producing mRNA in living cells. Can you explain what makes this approach different from the conventional method, which is in vitro transcription or IVT? And I'm not sure if our listeners are familiar with this term and how is it different from what you're doing. But why it could be a big deal, the making mRNA inside living cells and for future therapies.
So to understand this, I think we first need to really appreciate how the mRNA is made conventionally. And today, pretty much all the mRNA in the world, all the commercially available RNA, is made via this in vitro transcription, which is a cell-free reaction where you take the DNA template, you take a linearized DNA template, and put it into reaction with a T7 RNA polymerase conventionally, capping enzymes, nucleotides, and a number of other cofactors, so that you can run this reaction. T7 polymerase transcribes the DNA into mRNA, and then you get basically the soup of mRNA and DNA and other parts of the reaction, and then you purify this mRNA out, typically using something called oligo-DT, or basically, if you want to really purified RNA, you do it with chromatography-based methods. And now there is a couple of challenges with this, and kind of thinking from input to output. At an input, first and foremost, we see challenges with the raw material supply. The raw materials for mRNA production or contribute to the bulk of the cost of mRNA manufacturing. Today, if you go and ask contract manufacturing organizations how much it would cost, it can cost $500,000 to $750,000 per gram of clinical grade RNA. That is very expensive, and sources say different things, but 60 to 85% perhaps is the raw material costs, predominantly capping enzymes, which are proprietary, patented, etc. But this is not the only challenge. The other challenge is logistics, because for every new batch of mRNA, you need to again go and supply, GMP grade DNA, GMP grade nucleotides, GMP grade capping enzymes. Well, that's a challenge, especially because the supply chain for these molecules is not exactly overabundant. And importantly, for therapeutic applications, we see these biggest challenges at the output. And really, mRNA is very often, and this is the plague of in vitro transcription, contaminated by double-stranded RNA, which is highly immunogenic because our cells have receptors, predominantly TLR3 receptors, that sense this double-stranded RNA, and they are kind of freaking out because they think this is a virus. And this double-stranded RNA is really hard to properly purify from the mRNA, and contributes to the immunogenicity of mRNA. Then the other problem associated with that is, okay, actual immunogenicity, because the immunogenicity reduces your therapeutic window. I mean, mostly the undesirable immunogenicity. And the way people are solving this problem is really changing the uridines in the mRNA for modified base pairs, pseudouridines, N1-methyl pseudouridines, 5-methoxyuridines, etc. And there was even a Nobel Prize given in 2023 for basically modified base pairs and their effect on lowering immunogenicity. But there is a couple challenges with it. The challenge number one is the cost. And the modified uridines are significantly more expensive than natural uridines. The second challenge is kind of the IP situation around this. The leading mRNA companies are suing each other around the basically who owns patents for this and that, basically nucleotides. And finally, there was a recent paper basically showing that our ribosomes, our cellular factories for proteins that take mRNA and make proteins, can slip on these highly modified mRNAs, and basically then this can lead to production of a small amount of mistranslated, misfolded proteins that immune system then recognizes. And all of these challenges, therapeutic window, immunogenicity, double-stranded RNA, logistics, cost, contribute to the fact that we haven't really seen an mRNA therapeutic approved beyond infectious disease vaccines, which we think are the lowest hanging fruit, because you're dosing typically very small amount of mRNA very infrequently, and some kind of immunogenicity is actually fine because you need to activate the immune system. But let's take something like protein replacement therapy, which I think is the opposite spectrum. You are dosing significant amounts of mRNA, typically say 0.3 to 0.9 milligrams per kilo, which is roughly 500 to 1,000 times more per dose than the typical, say, COVID vaccine. And you are dosing this frequently, say every 2 weeks, and you don't want to have any immune activation. So I think that this is a big challenge, and we believe that our technology is addressing it.
That's amazing. Yeah, thanks for the summary. So you're saying basically it's cheaper, it's pure, it's more scalable, and then the immunogenicity problems can be solved with this way, basically. Is it fair to think of this as a foundational shift? Not just a more efficient process, but an entirely new category of mRNA manufacturing?
I think this is exactly the way to think about it, and I'm sure Krishna will speak more about the design, but this is not just about manufacturing. We think that what Sensible is building is the most differentiated and kind of integrated mRNA design, optimization, and manufacturing platform there has ever been. Basically, again, what our core thesis is that if you make mRNA in cell and design mRNA for cell-based production, because if you're making RNA cell-free, you need to make compromises. T7 polymerase is not exactly eukaryotic polymerase evolved to transcribe long eukaryotic genes, right? So basically, if you design mRNA for cells, and if you make mRNA in cells, which have all the necessary machinery to make high-quality RNA and to actually naturally modify it with the eukaryotic modifications, of which there are plenty, you can get mRNA that has reduced immunogenicity, has minimum double-stranded RNA, and can be designed for much better performance while completely eliminating some of the challenges of the supply chain and the cost.
Yeah, thank you. Thanks, Miro. Yeah, so let's get a little bit into the technology before we talk about the strategic partnerships, perhaps. So, as we said, we are engineering living cells to produce mRNA. What does that look like in practice? How do you design those cells, and what makes it a scalable system?
Krishna, do you want to have a go on this one?
Sure. I mean, for the design process, it's ours is we're actually starting from first principles here. And it's actually really interesting because we're kind of running into, as Miro pointed out, about traditional IVT manufacturing. They're they're kind of putting the cart before the horse in this case, right? We're trying to make something synthetically that's supposed to mimic what we see natively in our RNA, which isn't necessarily a viable process. So our design and manufacturing process is very tilted towards actual biological observable data that we see that's been collected throughout 30 years of science, but as well as our using our own internal data sets. So that design process is actually going in from design from say optimality of therapeutic window, but also reduction of off-target contaminants, in this case, like say as Miro said, double-stranded RNA, which is a big one in mRNA vaccines, as well as so that design process, when it's actually scaled up and we're producing it in living cells, we can produce that at high yield. I mean, and it's really scalable in this sense. This is actually a very robust technology. So the benefit of this, as Miro also mentioned, is that all 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 cells have native error proofing, so they will check for proper product. So we're actually leveraging Mother Nature, who's the best engineer in this case.
Mhm.
She'll make better products way better than any other than in any other situation. So this is a set of like essentially design, test, and iteration, and we can use this. And the marvel of this is that, and we're kind of in this watershed moment of RNA vaccine manufacturing, is that it's a new delivery mechanism. So historically, some protein therapeutics have failed, um, because of challenges in delivery, or actually in the challenges of penetration. Right? If you create a antibody, you can't necessarily deliver it to your actual affected region for various reasons that are complicated for each drug. Or, but in this case, we can design new therapeutics that can specifically target um, these specific uh, therapeutic indications. And so our benefit is that because we can scale up really quickly, we also have low immunogenicity, and so we can do this and treat it actually quite very difficult patient populations as a result of it.
Mhm. And then on the computational side, Krishna, how do you design and optimize the sequences themselves? I've read that your AI models simulate RNA folding and packaging inside the cells. So what does that involve?
So yeah, our so we use a mixture of coarse-grained molecular dynamic simulations with mixed in with some what's called full atomistic or full atom simulations as well, for essentially for prediction as for optimization as well. And then that's fed into our proprietary deep learning and transformer models that we've built in-house on our own data sets that can actually do this. And so the objective is to essentially do large-scale simulations and large-scale predictions based on for example, just minor or initial preliminary data sets. So the build test design cycle is going to be narrowed much smaller in this case.
So and then you recently got your design cycle right from 15 days to just one. What kind of modeling or prediction power made that possible?
So I mean, part of that definitely is there's been some great work that's happened recently um, on the MD side of things. There's been a lot of work because largely because of the watershed moment again of an RNA manufacturing. We've seen a much bigger focus on MD simulations on long disordered RNA structures. So there's been quite a few interesting pieces of research that have come out that have enabled this, as well as the the work that we've been doing internally, because we have our own internal QC data sets and QC metrics that we know are going to be set for specific optimality. So this kind of work is it's I want to say I want to take all the credit and especially it's kind of weird to say it because my boss is here, but I'm standing on the shoulders of giants here. Where
Mhm.
and I also want to reiterate is that the work that's happening is it's people contributing to the field in out of intense interest, but also there's a end goal, right? And so this modeling simulation work is really great, and there's been there is a mixture of my team that's put forward an incredible amount of work on this as well. So I want to take all the credit for that, unfortunately, but sorry, Miro, it's what we are. It's a team effort.
Yeah, let's go for it.
Yeah.
Yeah, we are a great team. A lot of super hardworking, super smart people. So it's a real pleasure working with Krishna and the rest of the guys.
Yeah, yeah. You build a powerful platform, cell-based mRNA manufacturing, computationally optimized in vivo. Now let's talk about how you're scaling it. So you just announced, well, a few weeks ago, really, announced a partnership with NVIDIA that dramatically accelerated your design optimization. As I said, from the 15 days to just one, but obviously that was enabled by NVIDIA. Can you walk us through what that collaboration looks like in practice? What do they help you with? What they bring to the table, and how has it changed your workflow?
Yeah, I can take this, Miro.
So maybe
Yeah.
Yeah,
Yeah.
maybe just let me give you a bit of a kind of elaborating on what Krishna said. Really, the big vision for our computational work, and then I will let Krishna elaborate on non-confidential bits of our work with NVIDIA. So really, the what we learned and what is really exciting is that cell-based optimization and design of mRNA for better cell-based therapies is really different than that for synthetic RNA, which really opens up this massive design space for us. And what we are building is really a super high throughput screening platform so that we can test hundreds to thousands of mRNA sequences at the same time and kind of correlate that structural characteristics to the functionality and functional outputs of the mRNA, and then use that for learning of the design rules for optimal mRNA therapeutic properties. That's really the big vision of super fast optimized design that then allows us to quickly make the cell lines making super high quality RNA that can quickly be scaled. And obviously, this requires a lot of resources, a lot of compute, a lot of data, and we are generating this absolutely unique data set. And before I will kind of hand it over to Krishna, I will just say that NVIDIA is absolutely amazing partner to work with. If you can, I recommend them to everybody. They are responsive, they are kind, they understand very deeply the core biology, and we had really amazing experience with them. So if they are listening to this, David, Cedric, Rene, Alice, and everybody. So yeah, but over to you, Krishna.
Yeah, I mean, and thank you. Yeah, I just um I'm kind of in awe with the NVIDIA team because they've done this in such a way that they've said that we've provided the workspace and we've provided the playground for you. There's you can you're the expert in your field. We can help you build from there. And they've got a lot of incredibly brilliant people who are working on this, who are and like that part of that time is that our speed up has been largely, I think, attributed as well to the support that NVIDIA has given us. Right, we can build accelerated machine learning models and AI models really quickly because they've made the ecosystem available to everyone. So our use case is so niche, right? There isn't that much viable data out there in the literature. So we can't really just say, oh, just go pull something out there and rebuild a model this way. We have to start from scratch or start from first principles. And that would be a daunting task if I had to, like go out and like literally just build simulation networks or do this kind of work that is it's not my expertise, and we'd have to switch into a completely different field to be able to do this accurately. So what we've happened is that we were invited to be part of the Brev program, which we got to see their high performance computing cluster, which is incredible. I am eagerly asking Miro to give me one of them for Christmas. So it's gonna be it's really the the challenge that we're working with is that is that the design space is so complicated. It is the primary data that we're coming from is something that is in either in IVT or something that is in orthogonal data that doesn't necessarily replicate to what we're seeing, and it's actually really challenging. So building out that infrastructure, which would have taken me months of work just on my own, is now possible. And I also want to shout out to the BioNemo team because they've made it so that they've taken commonly used public tools and optimized them so that things that say like things that would be things that would take forever just because we have to rely on academic researchers to do their work as well as maintaining code bases, which is obviously a challenge. We're expecting them to do this for free. NVIDIA stepped in and said we'll optimize this so that it'll be running more natively on our GPU architecture, which is really great because that means that I can pull some of these tools immediately and start getting working on at least testing. I know that I don't have time to do this infrastructure building, but especially because we're a startup, there's always 13 things on my plate. Miro's got double that every day. So this it's not, and I think I want to. It's not just the fact that this access to compute is a start. That's the starting line, and so we've had this is a very fruitful collaboration in our end because it's been incredible just because we've now leapfrogged in our capability. This is something that I presented Miro a roadmap in 2023, which I said like based on our existing infrastructure we won't be getting there until 2026. We're already there coming, it's been we're so far ahead just because of this.
So you mentioned that BioNemo, is that like a foundational model that you're layering on top of?
No, I mean we can talk about this later, I guess, in that sense. But they're essentially a set of frameworks that that NVIDIA has packaged and made it available. So they've made endpoints available as well. They're not just foundational models. Some of them are. So there's commonly, like say for example, there is the evolutionary scale. I forgot what the M is in the ESM, but ESM2, which is a very commonly used protein language model. There's other ones that have actually been optimized and put up onto this. There's commonly like something like GeneFormer, which I'm a big fan of for single cell work. So those have been set up and optimized. A lot of the work that NVIDIA also has done is they've put them into they've Dockerized or containerized their workflows and made them optimal to use on native NVIDIA architecture. So that's great. So you can just instead of having to worry about installation, you can just pull it and get started. And it's already up to date.
It's amazing. So you kind of touched on this a little bit, but what kind of design questions, I guess, are you now able to answer that wasn't really feasible before the collaboration?
So one of the things that we've definitely looked at is the challenge that we've worked with is RNA structure biology as well, right? So there is sets of optimization. So RNA designing new RNA is quite challenging in this case because we're looking at very little sparse data and we're looking at trying to ground truth, and we're kind of assuming that say for example this therapeutic is what we're seeing in nature, so it should be the best one. Not necessarily the case because we want to optimize against say double-stranded RNA adjuncts, which will happen sometimes. So this kind of thing, which would have taken us essentially, we would have to build out a large screening platform to be able to say answer that question. We can then say we can say okay, we found a set of optimal designs. We can now combine those optimal designs and say we can start bringing that in and saying we can test these instead of will this even work in our system? Will this even be scalable in our system? These kind of things that are really engineering challenges, not scientific challenges. We can leapfrog that.
That's awesome. Thank you. And then this month, I guess, or last week, you also announced a partnership with Sartorius to help scale your platform toward real-world manufacturing. What does that mean in practice? Are you preparing for any clinical trials or building bigger systems, or is it something else that sort of fueled the partnership?
So I hope that, first that Krishna didn't scare off every potential hiree by saying that I'm putting 13 things on his plate every day. But hey, you might get you might get a new high performance server for Christmas. So there is a balance. um, High performance in all aspects. Look, so to speak a bit about Sartorius, as a small company, one of the big challenges, if you really want to be a fully integrated company, is okay, we have made over the last 18 months 1,000 batches of mRNA, actually more than 1,000 by now, because our production team is moving fast. Now, we were searching for a partner that can help us to move towards kind of clinical manufacturing capacity as soon as we can. Because our vision is that our pharma partners come to us, tell us, we would like to have this mRNA or this protein designed, optimized, and ultimately manufactured. And we would be able to provide them with this designed, optimized, and manufactured proprietary mRNA made by a proprietary cell line in record time so that they can fully focus on preclinical development and ultimately on a clinical development. And really, we see ourselves as a therapeutic platform, and ultimately, we want to bring our own mRNA medicines to the clinic to help treat patients. And we found amazing partner in Sartorius, which is a global multi-billion-dollar company, which has an amazing mRNA arm. So they've been able to test our RNA, validate really its purity, for example, and they decided that they would like to work with us on scaling the mRNA, cell-based mRNA production, so that we can make enough for clinical trials and make it at a clinical grade quality. So we have internal talent to do this. Our Chief Technology Officer, Alan Prolong, has 30 years of experience of scaling biological processes. He was Vice President of GlaxoSmithKline for new technologies introduction and life cycle management. So he's super experienced. But as you know, this is an expensive endeavor. And if you can get somebody who has so many of the core tools for the process optimization, so much knowledge, and so much industry lead and industrial experience. I mean, you go to any pharma in the world and you say, "Oh, we are working with Sartorius on this scale up." They're like, "Okay, done." That's literally like, "Oh yeah, by the way, we are going to bring this so that we can make clinical grade RNA with Sartorius in 2026." And the feedback is like, "Okay, great." So I think that this is what many companies are actually, or many therapeutic platform companies kind of have been, damaged by not thinking about how they will bring up their processes to the appropriate scale and quality level. And we have been obsessing about this from the beginning because it's very easy to get super excited about how am I going to optimize, this 3' UTR and how amazing it's going to be. And the second thing, which is, oh, wait a minute, but do you realize that like supplying 2 milligrams of your RNA here, it doesn't really help me if I want to take mRNA to the clinical trial, and it will be hard for pharma companies to partner with somebody if they feel that, oh, but really, this is like 3, 4, 5 years from the clinic. And not because we can't run the IND enabling studies, but we just can't get a clinical grade material. And what does it matter what I can design if I can't make it? And I think that this is something we wanted to address. And we've been very privileged at Sensible that basically the leading companies in the field, from Ginkgo Bioworks in our earlier days, through NVIDIA, through Sartorius, and through multiple big pharma companies with whom we are having conversations, just started to see the benefits of the platform and thinking like, I can't say the name, but I'm kind of quoting a very senior executive from a pharma company who said like, well, guys, if this works out, you have completely changed this industry. And that's what we want to do. This is still risky, but if Sensible works out, this is going to be completely new paradigm of mRNA. And we really hope we will be able to bring dozens of new mRNA drugs to the
Yeah.
clinic.
Not if, when, Miro.
That's
Yeah.
right.
Yeah,
And
yeah, it's very exciting.
yeah,
Forward-looking statements.
yeah. No, and also the the other thing is that we're really excited about this partnership with Sartorius is that the and especially from the computational design aspect of this, there's quite a bit of orthogonal data that we need to be able to calculate, and so
Mhm.
these this is how we can do force multiplication. We can do multiple different tests, multiple different optimizations all in one go. Things that we had to do sequentially or in like brute forcing it is not really an issue now because the technologies now exists, and so this is great. There's, as Miro touched, there's what's referred to as the valley of death, which is in therapeutic manufacturing where people haven't talked about manufacturability, and so because our goal is to make any mRNA manufacturable, any therapeutic manufacturable. That means that we we can do this in ways that we combinatorially. So not just put something through, see if it works therapeutically, then it turns out, oh, we can't produce it. This way, so we can do this all in one go, all in one set.
And then you mentioned a little bit, Mira, about collaborating with other pharmas maybe down the road. But do you see yourself also making mRNA for your own therapy? So, or would that just be for sort of others to use your system?
Yes, we do. Really, my vision for Sensible is to be a fully integrated biotechnology company. We want to be able to go from design of exciting mRNA sequences all the way to bringing them to the clinic. And we are really looking to the companies like Biotech, which has really pioneered this field, or Alnylam, which, albeit more in RNAi, has also shown that you can build a platform company and make it in a standalone, get multiple products approved. And we just think that this is so incredibly exciting because I think that it's not that often that you can build really, really orthogonal biotechnology company where every piece of it is really unique. And obviously, we can't speak about our pharma relationships and where we are, but we are just very excited to see the interest. And something else that I would say, we see the interest from number of governmental and international organizations, right? We receive non-dilutive funding from NATO because they just literally think that this is a way to really protect the supply chain for mRNA. We are talking to multiple governments across the globe because they actually come to us and say, "Oh, this is the technology we would like to ensure," and we really believe that what we are building at Sensible can become a new blueprint for multiple new therapeutic venues and really the thinking about integrating from design to manufacturing. We've been the manufacturing innovation, and we think of ourselves again to the great extent as a design company, but we just think that manufacturing innovation is so much needed in cell therapy, in gene therapy, and I think that countries need to be thinking about how they will bring the manufacturing capacity locally because that was a big lesson or should have been a big lesson of COVID-19 pandemic that we can have uh, a European Union and this Schengen space and open borders and everything, but there was a global pandemic and suddenly we became confined to the rooms and to the small square meter footage, literally, not being able to travel. And I think that this is something that manufacturing innovation and building this resilience is really helping to resolve and can be pivotal for.
Yeah, so building on that a little bit, what kinds of therapies and breakthroughs do you hope your platform will make possible in the next few years?
Without getting into too much details just yet, I think that the areas where we want to move forward are the areas where our technology has the biggest edge. So if you're thinking about low immunogenicity, better therapeutic window, improved purity, this kind of clearly shows areas such as enzyme replacement therapies, genetic medicines, mRNA encoded antibodies or biologics. We think that these are basically some of the broad areas where our technology can be going for. But ultimately, I think Krishna very nicely put it a bit earlier. We want to be able to make any mRNA sequence at the desired quality so that we can make safe, effective, and affordable mRNA medicines. That's all we are caring about.
It's amazing. Maybe just a side question for you. What is your opinion on cancer-based mRNA vaccines? I know it's a bit of a hot topic now, but there hasn't been much going on, and people have maybe right after the pandemic they were placing big bets on mRNA cancer vaccines, but there hasn't been much happening.
Well, I would say this that I think I have really fingers crossed for BioNTech, Moderna, and others who are developing them because it's sorely needed. Now, as you can imagine, it's a really kind of cutting edge of technology, and it's difficult, right? How do you balance the immunogenicity to stimulate the appropriate cellular immune response versus how do you avoid some of the side effects? How do you make sure that the manufacturability or manufacturing of the RNA is fast enough? How do you make sure that this actually becomes affordable? How do we regulate it? And so Sensible is also based in the UK. We have a lab in Oxford, and I'm glad to say that the UK seems to be really a leading country on developing regulations for personalized mRNA cancer immunotherapies. So I think that there's a huge potential in applications of mRNA technology in oncology space, and I think we are seeing the first or some of the first beacons of hope. Uh, is the technology where we would like it to be? I think there are still many challenges to solve. We hope to be able to solve some of them at Sensible.
Yeah, I mean, um, it is also, I mean, it's only been 5 years, right? The first mRNA vaccine has been only been put out in
Right.
5 years. So it's it's a novel, it's a completely novel modality of delivery, and so there's a lot of biological challenges that we're going to face with, and we hope to solve some of them, as Miro said. But the we remain extremely hopeful for this because it's the it is the hopefully a new modality that's unlocked. So traditionally, a lot, well, not just a lot, but some cancers are quite difficult to treat. So this might be the avenue in forward. There's a lot of great um, talent and people working in this field, and I'm an optimist as this goes. It's only I like to think about it. It's like what when people say, "Where's the cure to cancer?" It's like, well, it's we come up with something that's completely new therapeutic vaccine in under 12 months. Give us a bit more time.
I would also say that I think it's a testament to where the mRNA technology is going that in the last 8 weeks, we have seen three major acquisitions of mRNA companies. BioNTech acquired CureVac, uh, and CureVac is building very exciting cancer immunotherapy portfolio, for example, for glioblastoma. Um, Eli Lilly acquired Verve, especially for their cardiovascular kind of hopefully one-off treatment to diseases like familial hypercholesterolemia. God, that's a hard word. And finally, a very exciting area in a CAR T space where AbbVie acquired Capstan for over $2 billion, and I think that there is just so much going on in mRNA space. Orbital Therapeutics just 2 days ago announced that they have seen very exciting and promising preclinical data on circular RNA for CAR T in autoimmune space. And I think that, you know, the future we are imagining with Mario and Krishna, Alan, and the whole of the Sensible team, and our investors and our advisors and everybody in Sensible Universe, is that there will be dozens, maybe hundreds of mRNA medicines approved in the next decades, and it will be as big of a revolution as really the biologics were a couple decades ago.
Yeah. Thank you so much. Yeah, this is great. So I have one last question really before wrapping up. But so, what is kind of the next big milestone that you're focused on, whether it's clinical, technical, commercial at Sensible?
So really what we are focusing on is advancing our collaborations with some of our prospective pharma partners, and we are really excited to see the interest on that front. On the technology side, we are very excited to see the progress on building the high throughput platform, and so huge kudos to Krishna and actually to our colleague Mario, who's been really pushing this work together with our senior scientist Calvin, etc. And we are really excited to see kind of closing it on both ends, really, the high throughput screening platform development on one side, pushing our process development and moving towards clinical grade capability by end of next year, hopefully. And finally, pushing these platform partnerships with prospective pharma partners. So I would say that these are the three core things: the platform, the clinical grade manufacturing, and the prospective partnerships.
Yeah, thank you. And good luck. It's really amazing what you guys are building and what you've done. Any final thoughts that you have that you would like to share, maybe?
So maybe I can start, and perhaps Krishna has some more to share. So first of all, what I'd like to say, and this is general ecosystem call, it's a tough time for life science, as many people know, but um, some of the most generationally important companies have been and had been built during the tough times, at the brink of crisis and at the brink of bursts of bubbles. And I'd like to encourage everybody to consider building a company, because it is hard, and it can be painful, but it's extremely rewarding. So I wanted to encourage all your listeners to your amazing podcast to go and build if they can. Uh, the second thing I'd like to say is that um, there's a lot of challenges to solve in biology, and especially in the mRNA space. So I'd like to encourage people to get in touch with us, and we are always happy to think about how we can work together, how we can learn together. It is a hard problem to bring new mRNA medicines to the market, and I think it ultimately needs to be a team effort of the community. So this would be some of the core kind of calls to action I'd have.
Thanks.
Yeah, thanks, Maro. And yeah, this is something that I feel very passionate about as well, right? The power of science to build a better world, and that's what we're all trying to do. Um, and it's important to think about how far we've come collectively, but also where we're going. And this is a transitional moment. This is one of the things that we'll look back 50 years from now and say, this is one of those times. Like, we're going to go back and look at, say, this, the invention of penicillin, how it changed medicine, how this is going to be one of those moments. And it's I'm fully convinced that the human spirit can conquer any challenges, any even if the laws of physics are against us, we'll conquer this collectively. So it's, as Miro said, and this is only really possible by us all coming together collectively. And so if there's anyone out there who is working on things that say, "Oh, can never work," come on, it'll work. Give it time, will work. And we're happy to talk to anyone out there who has a weird idea, who has a really insane idea.
And people told us this will never work at the beginning. Oh
Yeah,
boy, did we hear that? Like, this makes no sense. Like, why would it go back to cells if it's like already cell free? Like, this makes absolutely no
yeah,
sense. Like, why would you do this? And here we are. And by the way, there's still a long journey ahead of us, right? But now I would say that the skepticism is really muted. And yeah, and by the way, as you can see, we are determined bunch, at
Yeah.
Sensible.
Yeah.
So yeah.
Definitely.
It's when Miro interviewed me, my first thought was this was never going to work. The next day, I called him up and said, "This is no, this is going to work." So it's um it's what we call in Midwest elbow grease, gumption. We call that just just roll up your sleeves. I guess, well, in this case, roll them down because we have to be careful about proper PPE. But, but you know, put it. It's it's really remarkable, just what I'm seeing. Right? It's people come in, especially my entire team. I want to thank them out. I'll call them out especially because they've biology is a weighted random number generator. Sometimes it feels like you're playing the slot machines and always losing in some cases, but it's the work that we've done, the amount of challenges we've overcome collectively in the course of 8 months has been remarkable. And so that's reinvigorated my love of science and my my passion because something that we thought was people dismissed out of hand, we're now regularly doing, and it's not even exciting for us anymore because it's just a routine. This is a random Tuesday.
Okay, thank you so much. Samira, Krishna, thank you so much for joining me today, for sharing a look inside the future of mRNA manufacturing. I'm personally really excited to watch you, keep watching you and see you achieve great things. I think there's a lot of greatness ahead of you. And so for those of you listening, you can learn more about Sensible at sensible.bio. And if you enjoyed this episode, make sure to follow Biotech Blueprint for more conversations at the intersection of science, technology, and strategy. I'll see you next time. And thank you.
Thank you,
Thank
Catherine.
you.
Thanks.